Good afternoon. So today we're most fortunate to have as a Walsh lecturer Ulrike Heberlein. She has come a long way since she was the national high jump champion in Chile, her native land. She studied biochemistry at the University of Concepcion and then took her PhD under the mentorship of Robert Tijan at the University of California at Berkeley. This was followed by a postdoctoral fellowship with Gerald Rubin, also at UC Berkeley, following which she joined the faculty at the University of California at San Francisco, where she became professor in the year 2003. Since 2012, which is when she moved east, she has been the scientific program director at the Genelia Farm Research Campus of the Howard Hughes Medical Institution. There, she continues her exciting work, which we will hear about, using Drosophila as a model for studying alcohol addiction, and also has a major administrative role. She received many awards, and it just shows that these awards ultimately truly pay off. She received awards from the Howard Hughes Medical Institution, McKnight Foundation, and a Presidential Early Career Award for scientists and engineers. And in 2012, she was elected to the National Academy of Sciences. Ulrike is an academic editor of the PNAS and on the editorial board of Cell. She served on many NIH and international review panels, including being a member of the board of scientific counselors of the NIAAA. She's participated in many, mainly Cold Spring Harbor and other courses which link neurobiology, Drosophila genetics, and addiction. Her early research was on growth and differentiation in the Drosophila eye, but then in 1998, she began to explore the use of Drosophila in the study of addiction, particularly to alcohol. Taking advantage of Drosophila genetics and the fact that flies are virtual alcoholics, ingesting much ethanol from decaying fruit, she's characterized at the molecular and genetic level how flies develop tolerance and dependence on alcohol. Later, this work was extended to cellular mechanisms, pathways, and behavioral activities, and the interplay between neuroscience and addiction in general, alcohol in particular. Her innovative work, which has bridged these disciplines, has brought remarkable insight into mechanisms of addiction and tolerance, primarily for alcohol, but as applied to other species than the fly. Now, all of this and other activities don't deter her from admitting to being a baseball fanatic who, by coming east, maybe will bring good fortune to the Washington Nationals. Join me in welcoming Ulrike Heberlein, who will present a Walsh lecture entitled Drosophila as a Model for Alcoholism, an Interplay of Nature and Nurture. There will be a reception sponsored by FAES at the conclusion of the session.
Thank you. I don't think this is on. Is it? It's on now. Okay. Well, thank you very much, Wynn, for that incredibly gracious introduction, as well as for the honor of being able to give this lecture here in front of all of you. So, my lab just moved in the last couple of years from the West Coast to the East Coast. So I don't really have really great new data to show, so I decided I'm just going to repackage old stuff in a new way and see if I can fool people. So anyway, what I'm going to tell you today about is how we have developed Drosophila as a model organism to study drug addiction in general, but more specifically alcoholism or alcohol use disorders, as they are more correctly called these days. And I'm going to then tell you a couple of stories about the interactions of genes and experience in the developing of alcohol-related behaviors. So alcoholism. Alcoholism is a big medical and social problem, not only in our country, but in the world. And what sort of strikes me is that it doesn't just only interfere with the life or even end the life of the alcoholic, but there are many, many innocent people, bystanders, that are killed or are maimed or injured as a consequence of some people drinking too much. It is an expensive problem. It was estimated about 10 years ago that the cost to the US economy of alcoholism was about $200 billion, and that's only really what we can measure. So I would say it's probably a lot more if we were able to measure everything that's related to it. And it is a really difficult problem. And the reason that I think it's difficult, or people think it's difficult, is because, number one, alcohol is a very non-specific drug. It's not unlike other drugs of abuse, which have a specific receptor in the brain and act at, sub-micromolar concentrations. Alcohol has multiple targets in the brain. It acts at fairly high millimolar to tens of millimolar concentrations. And the challenge is then to figure out how it is that all of these somewhat non-specific and not very strong interactions lead to the changes in the brain that ultimately lead to addiction. The other difficulty with alcoholism, which is actually true for all addictions and many complex diseases, is that the genetics are complex. The genetics are both multigenic and heterogeneous, which means it's very difficult to identify genes that may confer risk for alcoholism. And since the genetic risk factor contribute only about 50% of the variance, you can imagine that the other 50%, which is gene by environment interactions, are equally complex. So finally, I want to tell you that alcoholism is actually a very old problem. Probably since the time, oh, this is much better, no light in my face. Since the times when primates first walked the earth, there was already fruit, fleshy fruit, and there were already yeast since the primordial soup. So essentially, our primate ancestors probably consumed a diet which had anywhere from 3 to 5% alcohol, for probably millions and millions and millions of years. And eventually people started growing certain fruits, such as grapes, for specifically fermenting alcoholic beverages. And alcohol was actually considered in the ancient civilizations the best thing that could happen. I mean, I think everybody probably lived in sort of a slight state of intoxication continuously, all from children to adults. And the reason was, well, I'll tell you the reason in a minute. And the Greeks and the Romans and the Sumerians, they had gods and goddesses of alcohol because it was such a great thing. However, it was already sort of 3,000 years ago, no, 2,000 years ago, that some very prominent philosophers, such as Plato and Socrates, realized that while alcohol was this greatest thing in the world, there were some potential problems. And public drunkenness was not unheard of in those cultures. So, but the bottom line is that in Western civilization, until the 19th century, alcohol-containing beverages were used as thirst quencher. Water was poisonous because, of course, it was crawling with nasty microorganisms that would kill people. And in contrast, in the Far East, people realized that just boiling the water to make tea killed all those microbes, and so there was much less actually alcohol use in the Far Eastern cultures throughout history. Is this hissing like I hear it, or is it okay? Hmm?
It's fine.
It's fine, thank you. That's a Drosophila melanogaster, and that's the model organism that we decided to use. Started about 20 years ago to try to understand both the genetics and the physiology related to alcohol. And at the time, it was a pretty crazy idea. But we took on the challenge, first at the Gallo Center and then at UCSF, and essentially decided, look, you know, Drosophila has been such a wonderful model system for many, many, many developmental questions, for many disease-related issues, that why not for alcoholism? So, as probably most of you know, flies are the premier model organism for genetics due to the work of T.H. Morgan and his colleagues at Columbia in the early 1900s. Indeed, much of what we know about genetics comes from work in Drosophila. Flies are easy and fairly inexpensive to raise in the laboratory in large numbers. So again, you can do genetics. Their generation time is short. Or did I already say that? Sorry. And I think most importantly, because it has been the model organism for so long, the amount and diversity of tools that exist to manipulate every single one of the fly's 16,000 genes, and almost every single one of its 500,000 neurons exist. And so it's essentially an incredible opportunity to be able to use these tools to understand the genetic and the neurobiological basis of alcohol use in flies. I should say 20 years ago when we started this, you know, the scenario, that was before the genome was sequenced. So, okay, so that as a matter of background. So very often when I go out and give talks, I'm asked, well, do flies get addicted? And I say, well, addiction is really, by definition, a solely human condition. But there are several features that are used to define alcoholism. So I'm going to take the liberty to be a little bit anthropomorphic for a while and try to convince you that maybe flies fulfill some of the criteria that are often used to diagnose humans as alcoholics. And then I'm going to tell you two stories. Sorry, all my slides are cut off at the bottom, and if I forget acknowledgement of anybody, it's because it's written right there, and you can't see it. So we have about, as I said, 50% of the risk factor is genetic, and 50% is somewhat environmental, experiential. I'm going to tell you two stories, one about a gene called happy hour, and one about how experience, in particular sexual rejection, affects ethanol consumption in fly. And that's sort of the nature-nurture story that I said I would talk about. Before anything, I want you to see what drunk flies do. So they're being exposed to vapor in these tubes, and they're kind of just walking around. After a while, they get a little hyperactive. They start jumping around. I guess I'll let you watch. They get hyperactive at a blood alcohol concentration that's similar to what would make us hyperactive, and sedated at, again, a concentration that's similar to what happens in humans. You can see they're starting to get quite uncoordinated, having a hard time righting themselves, a hard time climbing up the walls. So as you can see, an intoxicated fly is just like an intoxicated whatever organism, including human. So I would argue that intoxication is quite universal, and that perhaps the mechanisms, therefore, may also be quite conserved. So here are some features of alcohol addiction as defined in the Diagnostic Statistical Manual 4. Of course, this is a bit outdated by now. And I'm not going to read these right now because I'm going to go through them one by one. So the first is the development of tolerance. It takes more and more drug, essentially, to achieve the same effect. So this is relatively easily done in Drosophila. And one of the assays that we used early on to measure ethanol intoxication in Drosophila are these inebriometers, which were originally invented by Ken Weber. And they're essentially columns that contain 26 baffles on which flies can perch. So if you put flies into the top, they will stay there because they're negatively geotactic, they go up. But as you circulate ethanol vapor through the column, they sort of start falling from one of these baffles to the other. Eventually they come out of the bottom, they get counted, and the data fed to a very, very ancient computer. And so what you get essentially is a number of flies as a function of time. You get an elution profile for wild-type flies, and this also allows you to isolate mutants that are either resistant or sensitive. So for those of you that are biochemists in the audience, this is essentially drunk fly chromatography. But not only can we use this to isolate mutants, but we can also use it to look at the development of tolerance. So we take the elution profile of flies during the first pass through the inebriometer. We let them rest for 4 hours, and then we pass them through again. And as you can see, if we look at the mean elution time, which is about 20 for wild type, and it's about 28 for tolerant flies, you can see that there is a shift in both of these in both of these parameters, suggesting that fly develop tolerance. Well, I don't have time to tell you that we have isolated a series of genes that are involved in the development of rapid tolerance in flies and in mice, and we also have found through forward genetic screens in flies the fact that the neuromodulator octopamine, which is the fly homologue of adrenaline, is involved in the intolerance development, a fact that has been shown in rodents. So again, it seems that the mechanisms are conserved. So let's go to withdrawal symptoms. We haven't really done any of this work, but beautiful work from Nigel Atkinson and his colleagues at UT Austin. They have actually looked after chronic exposure to alcohol, and they have done electrophysiology and have found that the nervous system is in a state of enhanced excitability that is consistent with a withdrawal, and this goes away if the flies are administered alcohol. Now, use in larger amounts than intended, I don't think we can ask the fly what they intend to do, but we can ask if they show escalation in use. So let's take a look at that. Let me define another little assay that we use to measure ethanol consumption in flies. These are little tubes with capillaries. 2 of them contain regular food, and 2 of them contain food that has been laced with 15% alcohol. And then the flies, just a few flies, go in, and I'll show you a movie in a bit, and they drink this liquid food, and we can just measure how much they drink by the descent of the meniscus in these capillaries. And what Anita Devineni in the lab showed is that if you look at the consumption per fly per day, and these are the number of days, they increase their consumption of ethanol-containing food at the expense of the other food. And if you calculate a preference index, which is essentially this difference divided by the total volume, so in this case 0 would mean no preference, they don't care one way or the other, you can see that preference actually develops quite strongly over the 5 days of the assay, to the point where they drink 75 to 80% of their food from the ethanol-containing capillary. So clearly, they show escalation of use. And they will voluntarily drink to intoxication. So in this case, the flies are starved for a few hours prior to the assay in order to force them to be synchronized in their drinking behavior. And you can see that flies in 10 minutes will drink as much alcohol to give an internal alcohol concentration of 0.3%, which is severely intoxicating. And this is a movie that shows how these flies drink out of these capillaries. So they stick their mouth parts, called the proboscis, right in there. You'll see this fly is pretty calm, and after a while it starts kind of getting a little tipsy, running around in circles. Now the fly will come down. It's like, get away, this is my booze. And anyway, what's really funny is once this fly tries to go back up, you can see that it's actually quite severely intoxicated and has been a very hard time getting up there. There we go. Okay, so they show escalation in use. We can't possibly know if they try to cut down, but Carla Kahn, a former postdoc in the lab who now is at Brown, is pursuing a project to look at persistent desire. Effort is spent obtaining alcohol. We now have an operant assay for alcohol consumption in flies, and so we should soon know whether flies exert effort to obtain alcohol, and this is in the works. Finally, we're very interested in understanding what effects does alcohol have on social behavior in flies. And so this is a big new project in the lab at Janelia Farm that we're doing in collaboration with Kristen Branson, who is a computer scientist and has taught us how to do machine vision and machine learning. Relapse. So if we go back to exactly the same little assay, but now deprive the flies in red. So here we have the preference index. Remember, 0 means no preference. These are the days of the assay. And these are two groups of flies. They're treated identical. By the 5th day, they drink quite a bit. And this group was maintained on alcohol. The black group, I have no idea why there are these little jiggles here. But as soon as you deprive, essentially you change the alcohol-containing capillaries for regular capillaries, essentially the preference is completely eliminated. But what happens is they very quickly, as soon as alcohol is put back in there, they recover high levels of alcohol consumption, which is one of the characteristics of relapse. I'm not ready to call it relapse, but relapse-like behavior. And the same thing can be done after multiple days of deprivation. Finally, use despite adverse consequences. I'm going to have to move here. So this is, again, the same assay, but we are comparing 3 types of foods now. This is, okay, so ethanol controlled. Okay, so this is just the same I've been telling you. 2 of them have food, and 2 of them have food plus ethanol. Escalation over time. In black is the choice between food and food with quinine. And quinine is very bitter. Flies don't like it. In fact, they hate it. They show a negative preference index, and they never learn to like it over the 5 days of the assay. But if you put quinine in the alcohol food, even though they start hating it, over time, they develop preference for alcohol. So for some reason, the alcohol is overcoming the aversive effect of quinine in this particular assay. So the next assay that has to do with use despite adverse consequences, first I need to describe you an assay for conditioned odor preference that Carla Kahn in the lab developed. So you choose 2 odors where naive flies show essentially no preference for one or the other. And then you train flies. In this case, odor 1 is given for 10 minutes, and then the same flies get odor 2 plus ethanol, vapor. It's mildly intoxication. And the reciprocal group has odor 2 plus odor 1 plus ethanol. And then they're put in a Y maze with the 2 odors and asked, where do you go? And what Carla found is that if you look at the condition preference index, that they prefer the odor that had been paired with alcohol. And not only that, they remember it for 7 days. 3 10-minute exposures to alcohol lead to this extremely long-lasting memory. And this is essentially indication that they find ethanol intoxication rewarding. But let's get to the use despite consequences. Well, this is not exactly use. So what Carla did is she put a little electric grid at the entrances of one of the tubes of the Y maze leading to one of the odors, and then asked whether, if this odor had been paired with ethanol, are flies able to overcome an electric shock to get to the odor they think is associated will deliver ethanol. And so here we're looking at the flies that overcome the CS-associated shock, and this is the volts, 100, 120, 140. These 3 lines at the bottom are all the controls, and you see in red here, these are flies that have been ethanol conditioned, and they show a higher percentage of overcoming the shock relative to the controls. The same is true for flies that have been conditioned with sugar, which of course is another reward. So I think that although some of these things we can't possibly ever ask a fly to do because they're purely human, there are other things that we definitely can study in a fly, and we just have to be very creative because unfortunately flies won't talk back, but we have to develop assays that would allow us to really get a sense of what's happening in their brain. So that's the end of this part of my talk. I think in order to be diagnosed an alcoholic, humans have to have a positive answer to 4 of those questions. So I think the flies so far are doing pretty good, and I bet you in a couple of years this is probably going to go up to 7. So I think flies have flies show many behaviors that are associated with alcohol use disorders in humans, and certainly those that are seen in more complex model organisms such as rodents. Okay, so I don't know if you believe me or not that flies can become alcoholics. I'm not even sure myself, but we are working on this every day to try to make a stronger and stronger case by developing more and more complex behavioral assays. Maybe someday we'll teach the flies how to talk. Okay, so now I'm going to tell you about the story number 1, which has to do with the genetics. Happy hour from an unknown fly gene to a preclinical rat model. So this is sort of your stereotypical way you go about doing forward genetics in flies. And the approach is very simple. First, you observe drug-induced behaviors. You already saw those. You saw some of the quantitative assays that we use. And then you can do completely unbiased genetic screens, which means you make absolutely no assumption about what the genes or the mechanisms may be that are related to these behaviors. Once you have a mutant that has abnormal behavior, you can very easily clone, identify the disrupted gene, which provides candidate gene to be studied in rodent models and in humans, and of course they provide tools to get at the mechanisms. So I'm going to tell you about one screen that was done by a former graduate student, Amon Kaur, whose name is hiding right down there. He did this heroic effort. He took 2,000 strains of flies and tested them in a behavior that I'm about to describe to you. He isolated 40 putative mutants. After cleaning up their genetic background and retesting, he ended up with 8 mutant strains, and I'm going to tell you the story of one of those, a strain that he called Happy Hour. You'll see in a minute why this strain is called Happy Hour. So this is the assay. For some reason, this movie doesn't work. So there's ethanol vapor passing through these tubes, and essentially you roll the tubes every few minutes, and you ask how many flies that have fallen on their back fail to right themselves. It's a commonly used assay in rodent models for intoxication. And then it's very simple. You just measure the number of flies that are not sedated as a function of time. Everybody's sedated out here, and you calculate a time to 50% sedation. So you're going to see some ST50s, the time to 50% sedation, which for this wild-type strain is about 12 minutes. So happy hour compared to the control in gray, these are two different alleles, mutations in the same gene, are very resistant to the effect of alcohol. In fact, they have SD50s of 20-something minutes, which is by our standards pretty robustly resistant. Oops, again cut off. Well, Ammon, having these two transposon insertions in the 5' end of the gene, he cloned the surrounding regions and queried databases and found that happy hour disrupts this gene, which had never really been studied, which has a kinase domain, N-terminal kinase domain, and a protein-protein interaction domain in its C-terminal. And this gene, which at the time was just a CG number in the genome, is really part of this very large family of sterol 20, the sterol 20 superfamily of protein kinases. And based on, I think it was just one paper available in the mammalian literature, it was believed that it's most homologous to this GLK, and that this ultimately would activate one of the MAP kinase pathways, the junk pathway. So Ammon set on to figure out, okay, what does happy hour do in flies? So we have receptor tyrosine kinases and other signals that essentially ultimately signal to these MAP kinase pathways. Again, we're missing stuff at the bottom. And the three canonical ones are the ERK pathway, the junk pathway, and the P38 pathway. And now I'm going to summarize 2 very frustrating years of Ammon's life. He tested every mutation in this pathway, nothing. Every mutation in this pathway, nothing. And then I said, but Ammon, why don't you test that one? Well, and sure enough, it turns out that the ERK pathway, which is very well conserved in Drosophila, and many actually of the components were first identified in Drosophila, and particularly downstream of the EGF receptor, seems to be responsible for the phenotypes. So I'm just going to show you very briefly here. So this is a cartoon of, here's the EGF receptor down to the MAP kinase. This ligand is called Spitz. So if you activate this pathway by overexpressing the ligand, overexpressing the kinase, or having a constitutively active MAP kinase, it all leads to resistance. Okay? So activating the EGF receptor pathway makes flies resistant to alcohol, which is the same you see with loss of function of happy hour. Conversely, any manipulation that reduced the function of this pathway, including manipulations of the EGF receptor itself, made flies sensitive to alcohol. So resistance is associated with EGF receptor pathway activation and a reduction or mutation in happy hour. Sensitivity is associated with inactivation of the pathway and increase of happy hour. So therefore, one would have to postulate that happy hour functions normally as an inhibitor of the EGF receptor pathway. So when you take it away, the pathway functions better and produces resistance to ethanol. So we don't know exactly yet what pathway is true, whether the EGF receptor inhibits happy hour, which inhibits MAP kinase, and that leads to ethanol resistance, or whether it is in parallel. But we have quite a lot of genetic and biochemical evidence that happy hour acts normally to inhibit the EGF receptor pathway. Okay, nice and good. A gene nobody had studied before, it somehow inhibits this very, very, very well-studied pathway. So we decided, let's just do some pharmacology. There are lots of EGF receptor inhibitors that are out there, FDA-approved drugs even. So we were able to get these 2 drugs from Genentech, erlotinib and gefitinib. I don't know why they give them these names. And Karen Berger fed them to adult flies now, and then looked at what happened to their sedation. And indeed, inhibiting the EGF receptor as predicted by the genetics with both of these drugs led to a reduction in an enhancement in sensitivity. So the SD50 is shorter. It takes less time for the flies to become intoxicated. Now, the beauty about these drugs is that they are FDA-approved drugs, particularly Tarceva is a drug that is commonly used in some cancers, but it's extremely well tolerated. So we next went to ask, well, what happens if we do this in a mouse? And again, we did this very simple loss of writing assay. These mice, oh, where's the needles? These mice received a sedating dose of alcohol and then increasing doses of Tarceva, and Jana Lim, who did these experiments, asked how long does it take for these mice to wake up? And you can see that at increasing doses of Tarceva, they take longer to wake up, which means, again, they're more sensitive, just like the flies. And the pharmacokinetics are normal. But most importantly, in collaboration with Jeffrey Sims and Selena Bartlett down here, we were able to get enough Tarceva from OSI to do experiments in a preclinical rat model. So this is a 2-bottle choice where rats get a chance to either drink water or 10% ethanol. And after a while, they drink quite a bit of 10% ethanol. And when Selena and Jeffrey injected increasing doses of Tarceva, there was a substantial decrease in the alcohol intake of those rats, while the sugar intake and the water intake was left unchanged. Tarceva also reduces lever pressing for alcohol. So this is, I think, in a very unusual story in which we start with a completely unbiased genetic screen in flies. We have no clue what's going to come out of it. We end up with a gene, happy hour. Well, first a mutant, then a gene. The gene was put through a lot of effort into a signaling pathway by using genetics. This signaling pathway provided us with a potential therapeutic target, and we validated this in a rodent model of addiction. So, you know, I was dying to get this patented at the time so that some human studies could be done, but that never happened, so it's probably never going to happen. But anyway, it's something that would be worth pursuing. Last story. Now we get to, you know, flies, sex, and videotape. So the second story deals with sort of the nature and the environmental and experiential part that affects alcohol-related behaviors. And we were thinking in the lab, what kind of really negative experience could we expose flies to? We thought about aggression and make losers and winners, but we didn't really know how to do that. It turns out that there's a very well developed assay, which is called courtship conditioning, developed in the 70s in Jeff Hall's lab, in which it's well known that if you put a male with a virgin female, they mate like this. If you put a male with a previously mated female, she actually rejects him quite vehemently. She kicks him, she runs away, she extrudes her ovipositor right in his face if he's trying to copulate. And so what we decided to do is to use this paradigm potentially as a way to generate flies that are genetically identical but have undergone two completely different experiences. Some of them got to mate with virgins, and some of them got really rejected and never got to mate. And then this happened for 4 consecutive days, this cruel or, you know, nice treatment, and then they were put into our ethanol consumption assay. And to our shock, it actually worked. So we're looking at the preference index here. Remember, 0 means indifference. And what you can see is that the flies that made it start with a negative preference index, so they avoid the alcohol. Then over time, you know, they increase the preference like all flies do. But the rejected guys start out drinking more, and they drink quite a bit more throughout the time of this assay. And I'm not going to show you all the controls actually published. This is the work of a postdoc, Galito Fear. And so then the question was, how could this experience translate into this change in ethanol consumption. And here is where Galit made an incredibly insightful, educated guess. She said, look, MPY, neuropeptide Y, in its fly homolog neuropeptide F, which are functionally and structurally homologous, MPY has been implicated in a series of negative experience. For example, soldiers that come back from war with PTSD have reduced MPY levels in their CSF. MPY levels are changed in rodents that are subjected to stress, such as maternal separation and physical restraint. And believe me, all the literature is down there. In a completely different group of investigators had found that MPY, this is Todd Thiel's group, affects ethanol preference in mice, ethanol sensitivity in flies. This is from, and Steve McIntyre, even in C. elegans. So we then questioned, is it possible that the transducer between this experience and the behavior is MPF? And if it was MPF, we can make two fairly simple predictions. The first would be is that rejection or mating affects the level of MPF, or the activity of the pathway. And we can measure that. And the second prediction is that the levels of MPF, or the activity of the pathway, should control ethanol drinking, right? So experience changes the level of MPF, and the changed levels of MPF drive increased drinking. So again, this is an experiment that Khalid Ophir did. This is looking at by qPCR of MPF RNA. This is the level in virgin males. It's reduced and rejected. It's enhanced and mated. And it's very obvious if you look at immunohistochemistry with an MPF antibody. So it's a brain of a mated male, and this is the brain of a rejected male. And this difference is greater than twofold. So clearly, MPF levels are inversely related to ethanol preference. Rejected males have high ethanol preference and low MPF levels, and mated males have high MPF levels and low ethanol preference. So the question is there really a causal relationship now between MPF levels and ethanol preference? So because this is flies and we can do genetic tricks, we could technically take mated flies, reduce the activity of the MPF signaling, and they should be transformed into flies that behave as if they had been rejected and show a high ethanol preference. Conversely, we can do the same with rejected flies. We could increase the activity of the pathway, and they then should behave as if they had mated. And again, this is something that's wonderful with flies, is that we have all of these tools. We use the Gal4 UAS system, so we express this yeast Gal4 transcription factor in all neurons, and we drive a double-stranded RNA that targets the MPF receptor for RNAi. So we're essentially down-regulating the MPF receptor wherever it is expressed in the nervous system. And we've looked at this in mated males. So mated males, remember, don't drink much. And in fact, our genetic controls don't drink much. They start out here and they move up. However, the experimental flies in red, in which the MPF receptor is downregulated, they show the same pattern of drinking as if they had been rejected. And the same is true if you do the converse experiments. So in summary now then, we know that rejection through some mechanism that we would very much like to get our hand on reduces the levels of MPF and thus the activity of this pathway, and that leads to increased ethanol consumption. Mating, on the other hand, increases the levels of MPF and thus activity of the pathway, and this blocks ethanol preference and consumption. So this is an example, I think, in where we now have encountered a mediator between a social experience and a drinking behavior, where this mediator is sensitive to the social experience and it's causally related to the changes in behavior that occur after the social experience. So I am ending here, right on time. I hope that I at least partially convinced you that flies are only going to get better with regard to being a valid model system for alcoholism. I told you one story how you can use flies and the power of the genetics in flies to do unbiased screen, end up with a gene that you have no clue what it does, and end up with a potential preclinical rat model for alcoholism with an FDA-approved drugs. And then I showed you, this is something we're studying more and more. We're looking at aggression as well as other social experiences now. And we're starting to understand the molecular mechanisms through which experiences in the environment affects ethanol consumption as well as many, many other behaviors. And I really apologize that the bottom of my slides were cut off, but these are the many people, past and present, who contributed. And the people that I have surrounded are those that particularly contributed to the story that I told you today. There are lots of postdocs, technicians, former graduate students, lots of kids, a wonderful, wonderful lab, and I feel very, very fortunate to have had these wonderful people in my lab for all these years. And finally, I'd like to thank you for your attention, and I'm happy to take questions.
Exciting presentation. There are microphones in the aisle. If you would please go and ask your question.
Beautiful story. So I'm curious, is it obvious that rejection of mating is only social experience? So if you would experience, because in principle it can also give some biological signals by releasing the material. So it's maybe not only social. So is it possible to reproduce something like this, for example, using this electric shock procedure? So in which case, if you will get the same result, that probably would strengthen the connection.
You're right. We have preliminary data for aggression that seems to go in the same direction. But I agree with you that it could be something that is deposited on the flies. They, of course, have cuticular hydrocarbons. We have measured those. There's absolutely no difference between them. But, that doesn't mean that their receptors could not be changed or, Right. So this is a very good point, and I think this is an area that we need to investigate more.
Thank you.
Please.
Very interesting talk. Thanks so
you.
much. I have a question about the data with the FDA-approved drug Drosophila. So the data with rodents do show that there is a sort of specificity which goes beyond potential sedative effects. It was unclear to me if the data with the Drosophila also suggested that the drug is working in a sort of specific way as opposite to a more sedative, unspecific effect. So if you can comment on that.
Well, there is a very good correlation in essentially every model system and in humans. This is work from Mark Schacker and the Koga group, showing that individuals that, for whatever reason, are more resistant to the effect of alcohol are more likely to become alcoholics. The converse is true for individuals that are more sensitive. So because we don't have these complex assays, or we didn't have them at the time we did those experiments, we just looked at ethanol sensitivity. And essentially, it was mostly to validate that the drug is doing what we think it's doing. It's actually inhibiting the EGF receptor in the adult fly and in the adult mouse. And the more complex, two-choice drinking behavior, of course, was then done in drugs. So, you do what you can with every model organism, the best you can, and then you ultimately get to. So they also do less lever pressing for alcohol. So there's clearly an effect here. I don't know. Those concentrations did not seem to have any really deleterious effects on the rats. So
Thank you.
maybe somebody will pick it up someday. when it goes off label.
Thank you for a great talk. I've been following this story since the early days, and it never ceases to amaze me what you come up with.
Thank you.
I had a question on, would you like to speculate on the mechanism by which rejection regulates NPY? And a related question, presumably most males aren't perpetual losers, they eventually get lucky. And the on-off rate, the plasticity of that, and if it eventually becomes permanent, or are there enough bad experiences that there's a threshold by which it can't go back?
Oh, that's a complex question. Okay. Do we understand anything about how the rejection experience, where are you? Oh, there. Okay, that's right. Affects NPY levels. You know, we have a suspicion that it goes through dopaminergic systems because some dopaminergic neurons are presynaptic to the MPF neurons. We are in the process of testing that, but that's total speculation, right? And so with regard to how transient the experience is, it really depends on how hard you train them. Right? So if you train them 3 times a day for 4 days, these males essentially don't mate for quite some time. Although, quite honestly, there are times a year, I don't know, there must be aphrodisiacs in the air, but some of the males mate with the rejected females, and we have to throw out that, you know, that fly because it no longer conforms our criteria. But maybe there's one thing I can tell you that may answer your question, which is if you take the rejected males and put them together with a large amount of virgin females that are aged enough that they're very eager to mate, they will mate. And it reverses the alcohol consumption back to baseline. So it clearly has to do with the act of mating more, I think, than it has to do with the act of being rejected.
Some of the alcohol intoxication manifestations in man are due to acetaldehyde. Do flies form acetaldehyde, and could that participate in some of the behavioral changes that you see?
That's a very good question, actually. There is growing evidence that so ethanol gets oxidized to acetaldehyde, which gets oxidized to acetic acid. And flies do it just like we do it with alcohol dehydrogenase and then with aldehyde dehydrogenase. And there is growing evidence that the aldehyde may be even having some rewarding effects. I'm not sure yet, but those are things we could test in flies because tools to manipulate those enzymes exist. But on the other hand, if you have too much of the aldehyde, right, if you have an active ADH and an inactive aldehyde dehydrogenase, you accumulate the aldehyde, which is very common in many Eastern cultures, and you know you get the flushing syndrome, which is accumulation of the acetaldehyde.
So, are there any? Oh, over here. Are there any genes linked to NPY which are affected by alcohol?
Actually, NPY itself is activated. It's activated by alcohol.
Okay.
Right, because that's sort of the converse. So it's a good experience, elevate NPF levels. Well, alcohol does also elevate NPF levels. We haven't looked at other experiences, but that we should do that.
Thank you.
It's funny, the sound came out of there.
So
okay.
The experiments, the mating experiments, is this a question of attraction between the male and the female, or is it fulfillment of some interaction? In other words, was Shakespeare right? Does alcohol increase the desire, diminish the performance, and promote the flow of urine?
Can I abstain? Call the fifth? Well, you know, so our flies are not intoxicated at the time when they are mating, right? So they go through the rejection process or the mating process prior to being exposed to alcohol. But believe me, there have been quite a few people in my lab who have looked at mating in intoxicated flies. And actually there is a paper published from Kay Han, who at the time was at UPenn. And her lab showed that if you expose male flies intermittently to alcohol, like we did in our tolerance experiments, that in the presence of alcohols, there would be what's called homosexual behavior, which is males start trying to court males very, very vigorously. Nobody's ever been able to understand why that is, but there's clearly a pretty strong effect of alcohol on courtship.
I have one question about whether alcohol has any effect on sex pheromones, either produced by males or females.
Unfortunately, I don't have a clue.
Thanks.
All right, if there are no other questions, Ulrike, thank you very much, and we will now adjourn for happy hour in the library.
Well, thank you very much, everybody. Great questions.